Okay, sir. Good afternoon to my respected professors, my seniors, and my fellow PGTs. Myself, Shobik Das. I'm pursuing my PGT degree in the Department of General Medicine, Bakura Shammilan Medical College and Hospital, Bakura. So, the topic of my presentation today is T cell versus B cell relative significance in immunity. See, first I will start with some definition. The definition of adaptive immunity is it is a silent response to the presence of infectious microbes by becoming active, expanding, and generating potent mechanism for neutralizing and eliminating the microbes. Now, I will touch upon the components of the adaptive or specific immunity. It consists of the antibodies, which are the immunoglobulins of 5 classes. Then comes the lymphocytes, which are of mainly B and T cells. The T cells are of 2 types, cytotoxic or CD8 plus T cells and helper or CD4 plus T cells. Then comes the role of major histocompatibility complex, which are the cell surface protein, and these are of 2 types, MHC class 1 and MHC class 2. Then comes the antigen presenting cells, which are most commonly are macrophages and other types of dendritic cells and B lymphocytes. A great role is also played by the natural killer cells, and the antigen receptors are the parts that are present on the plasma membrane on B and T cells. Now I will say about the antigen presentation technique. That the pathogen enters the body, which are first engulfed by the antigen presenting cells. Then the APC is fused with the lysosome to form the phagolysosome. The pathogen is then degraded into fragments of the antigen and then expressed on the surface of the APC with the help of MHC class 2 to form the MHC class 2 antigenic peptide complex. Then the helper T cells bind with the MHC class 2 with the T cell receptor. Then the APC releases the interleukin 1, which attaches with the other T cell receptors of the T helper cell. Then the T helper cell is activated, which on the other hand releases interleukin 2, which acts on itself, and then ultimately proliferation of the T helper cell into multiple antigen specific T cells occurs. Now comes the B cell proliferation. First, the pathogen is attached to the IgM antibody expressed on the surface of a naive B cell. Then the cell engulfs the pathogen, degrades it, and it is also presented on the surface of the B cell by the help of MHC class 2 by the same mechanism. Then the T helper cell is attached to the MHC2 complex, activation of the T helper cell. Then it releases interleukin 4 and interleukin 5, which ultimately causes the proliferation of B cell into plasma cell and memory B cell. Then the plasma cell secretes the antigen specific antibody, which bind with the similar antigen to form antigen antibody complex, which ultimately leads to opsonization. And then the antigen antibody complex is killed by the natural killer cell or destroyed by the complement activation. The mechanism of cell-mediated cytotoxicity is that first activation of the T helper cell occurs, there, which activates the cytotoxic CD8 plus T cells, which binds with the specific antigen presenting cell with T cell receptors. Then the cytotoxic T cells secrete cytokines like perforin and granzyme, which ultimately causes the destruction of infected cells. I will also talk about the relativity or the relative difference between the humoral and cell-mediated immunity. The humoral immunity, which is mediated by somehow T cells, mainly by the B cells and the macrophages, whereas the cell-mediated immunity is mediated by helper T cells, cytotoxic T cells, natural killer cells, and macrophages. The humoral immunity mainly acts on the extracellular microbes and their toxins, capsulated bacteria, and suppurative organism, whereas the cell-mediated immunity mainly acts on the intracellular microbes such as virus, bacteria, parasites, fungi, and also tumor cells. The humoral immunity recognizes unprocessed protein or the unprocessed antigen, whereas the cell-mediated immunity antigens are processed and presented by MHC complex. The Mechanism of humoral immunity is rapid, whereas that of the cell-mediated immunity is a delayed type reaction. Here, in the humoral immunity, plasma B cells,
শিবেন্দু দা আনমিউট করুন
plasma B cells secretes antibody. Here, the T cells secrete cytokines. Ultimately, I would like to touch upon some T cell related disorders like SCID, Digeot syndrome, combined immunodeficiency, hyper IgE syndrome, and also HIV and AIDS, where the virus infects and destroys the helper T cells, which impairs the ultimately activation of the cytotoxic T cells and B cells. I will also touch upon some B cell related disorders like X-linked and autosomal recessive agammaglobulinemias, hyper IgM syndrome, and CVID. I will come to my conclusion that cell-mediated immunity and humoral immunity both are important defense mechanisms of our body, and both of them act synergistically to protect us from infectious pathogens and other harmful cellular components. Thank you, sir.